Okay, so now we're going to discuss some different structural abnormalities with some examples. And so we're going to talk about balanced versus unbalanced structural abnormalities. So a balanced is just simply a rearrangement or some change in the location of genetic information. So we have rearrangements. And usually with a balance, that means no overall loss or gain of information. An example of these are going to be an inversion of a gene or a segment of the chromosome or a translocation. Now, an unbalance is going to be where you have material genetic information that will have a net gain or a net loss. Oops. Okay, so these are going to be typically associated with deletions or duplications. Chromosome breakages of chromosomes are going to have the ability to occur during meiosis, but they also can occur during mitosis. Usually they undergo some sort of repair if they do have some sort of chromosome breakage, but they can remain and alter the chromosome structure. So some structural rearrangements, again, unbalanced, we're going to have deletions. Deletions can be terminal at the ends or it can be interstitial in between. We can also have duplications, adding additional information. You can have the development of acentric fragments, and oftentimes those acentric fragments will lead to satellites that you lose, so you lose genetic information. And you can also have isochromosomes, fusions of chromosomes, so we'll talk about those. We also have balance, which is going to be translocations. There are a few different. There's a reciprocal translocation. There's also something referred to as a Robertsonian translocation. For balance, you can actually have an insertion, so you take genes in one area and move it to another chromosome, or you can have an inversion of those genes, but there's really no loss of the information. So an unbalanced structural abnormality is going to be a deletion. So again, this is where you have a loss of genetic material. So genes are lost. These can have severe effects. An example of this is the Cri-du chat. And this is where you have large numbers of genes that are going to be affected. A few others are going to be your Angelman, Prader-Willi syndrome are examples of these. And again, you're losing the material, so this could be caused by haploinsufficiency. You only have one good chromosomes for those materials, and it just depends on the genes that are lost in the chromosome that it occurs on. So a terminal deletion, again, is going to be at the ends, so the telomeres in this case, and then an interstitial deletion is going to be in the middle, sort of what we saw for the Cri-du chat and the Prader-Willi and Angelman. The terminal deletion is going to be a loss of those telomeres, and so that, of course, can have an effect on cells that have that deletion because telomeres help to protect the cells from losing genetic information. You also have, of course, haploinsufficiency. And this means that in this area where it was lost, the other chromosome cannot perform the function of two copies of the same gene. So you lose your product, and therefore you don't have enough of that protein, and it leads to a phenotype. Now, of course, deletions are going to depend on size, how much of the chromosome gets deleted, what are the genes in that region, etc. And the deletions can be caused by unequal crossing over, and this is going to be with misaligned homologous chromosomes. And it also can originate by chromosome breakage. So you can have chromosome breakage. That can be through UV radiation, other types of breakages, or you can have a loss of an acentric segment. So all of these lead to deletions and a loss of genetic information. So an example of a deletion is the Cri-du-Chat syndrome, or the cry of the cat. This is going to be seen in a relatively large amount of births, 1 in about 50,000. It's on chromosome 5. I believe it's highlighted here. This is chromosome 5. So you can see that it's missing the upper arm of chromosome 5, so the P arm. And so this will lead to the disease, and it has, even though children have the cat sound, it leads to a malformed larynx. That's what causes that. And you also have a mental disabilities, normal lifespan. Heart disease, and a mental disability. Another deletion-associated disease is something called Wolff-Hirschhorn syndrome. This is the deletion of the short arm of chromosome 4. And this would be the P arm distant. And so this deletion is only 2 megabase pairs from the telomere. So you're deleting your telomere, and you're also deleting 2 megabase pairs, and that is enough for this syndrome. In this syndrome, you have a cleft lip, typically wide-spaced eyes, delayed growth and development, some intellectual disability, as well as seizures. You also can, for this small, short portion of the chromosome 4, can also lead to muscle weakness, which has been resulted in increased lung infections associated with this syndrome. Now another is a deletion of your chromosome 11 in the P region, and this can lead to Wilms tumor. Wilms tumor is caused by the WT1 gene, and this is a zinc finger. And so this zinc finger is a transcription factor, and this is going to be important in growth, as well as kidney development, and as well as your gonad development. There's also absence of the iris. Sorry, I have to deal with my... Okay, so, sorry, my dog, is trying to come in here. Anyways, okay, so there's also anadrenia, which is absence of the iris. There's also genitourinary abnormalities that can occur with this deletion in the 11p region, and also some mental disabilities known as the Weiger complex. So the Weiger complex, it gets it from the Wilms tumor as well as the anhidritia, and then the genital urinary abnormalities. And the R is for mental retardation or mental disability. So that's what you get this from. So we have the W for the Wilms tumor, the absence of the iris, genital urinary disabilities, and then the mental retardation. And so this is, again, associated with the 11p chromosome. There's a microdeletion with respect to Prader-Willi or Angelman syndrome, so 70% of Prader-Willi are due to a microdeletion. And these are going to be a paternal chromosome 15q. And remember also imprinting of maternal. And this leads to the disease because you don't have the paternal genes present that should be expressing the genes, and the maternal genes that are present are imprinted and cannot be expressed either. Angelman syndrome, also microdeletion of the maternal chromosome 15q, so this is maternal. And then imprinting of the paternal. Also, Williams syndrome is a microdeletion with the elastin gene. So this is the ELN, or elastin gene. And also another gene, LIMK1, which has a brain-expressing kinase. And then these are going to lead to defects of cognition. And so you're going to have intellectual disability associated with these, as well as supravalvular aortic stenosis, pulmonary stenosis, hypercalcemia, etc., associated with Williams syndrome. There are also areas of subtelomeric rearrangements. And so we have here on chromosome 1, the P arm, position, your band 3, position 6. You have a monosamine in that region. And so this isn't the whole chromosome 1 is missing, but it's just a portion of that. And this is going to lead to distinct facial features with respect to this deletion. You have horizontal eyebrows, deep set eyes, the pointy chin. It's the most common deletion of chromosomes, so this is the most common. It can lead to severe intellectual disability, seizures, hearing impairment, as well as heart defects. So for identification of the subtelomeric deletion, you can use the FISH analysis. And again, FISH analysis can allow you to see smaller deletions. And basically, just as we talked about before, you're going to label your patient DNA with one color, and then you're going to label your control DNA with another color, and you're going to allow them to hybridize. And then you probe your subtelomeric regions, and then the probes will bind to the DNA or the chromosomes and will sort of light up. And then you can see whether or not the patient has hybridization of the probes. If a region is deleted, there's going to be a deficiency of hybridization. If it's duplicated, you will have excess hybridization of the probe. So it'll be brighter in the patient if it's duplicated, and it'll be dimmer in the patient if it's deficient, for example. So we talked about uniparental disomy already, and 30% of Prader-Willi syndromes are caused by this. And so this, of course, is when one parent contributes 2 copies of a chromosome and the other parent does not contribute a copy of the chromosome. So the 2 copies of one homolog is called isodisomy. So this would be the isodisomy. And then you have also 1 copy of each homolog, which would be the heterodisomy. So the disomy is important because, especially if a gene is imprinted, because if you only get 1 copy from, or both copies from one parent, and those genes are imprinted because of that parent, then that can lead to the disease because you don't have a copy of the unimprinted gene or chromosome. All right, so then there's also your duplications. So duplications, of course, is going to be a gain of information. So this is gaining information. And so depending on the genes, again, in this location, you can have excess gene products made. So this is referred to as a partial trisomy. Duplication can be caused by unequal crossing over during meiosis. And our X-linked color vision, as well as Charcot-Marie tooth disease, are both caused by unequal duplications. Okay, so for duplication, you have, again, 2 different, I'm sorry, that's translation. For duplication, the unequal crossing over is going to be between either homologous chromosomes, sister chromatids, and they can be duplicated or deleted. And so this picture actually shows it a little bit better. So here we have our homologous chromosomes. So we have our ABB, and then we have our C region here, and then we have our ABB and our C region here. And so you basically are swapping these C regions, but you're expanding on one chromosome the B regions, and you're losing that on the second chromosome. And so this is caused by unequal crossing over. And so you get a chromosome imbalance. And this can also, depending on where the deletion or the cuts occurred, so these obviously have to have some sort of splice in them. So it can disrupt genes. So it can lead to what's referred to as a phenotypic abnormality caused by disruption of genes. So in a particular disease, Pallister-Killian syndrome, it's where you have a duplication of the entire chromosome 12p region or a portion of the chromosome 12p. Okay, so another thing that can happen, which would be unbalanced, is when you form a ring chromosome. So a portion of a chromosome forms a ring when it has broken off and it has what are called sticky ends. And so the sticky ends like to attach to each other. So here we have our A and here we have our B, and so they come together and they anneal with sticky ends. And you still have here your centromere. Your centromere is still there. But what you're missing are, of course, your telomeres. So you lose your telomeres. And so if this will be called acentric if you lack a centromere. And then acentric rings are not going to be evenly distributed because there's no centromere. They can't be segregated equally into daughter cells. Ring chromosomes can also be caused by radiation, causes breaks, and then the breaks form a ring. So a ring chromosome missing a centromere would be, for example, if the break occurred here. So here this is C, oops, sorry, with this C, and then this would be D, and again you have your sticky ends, and so you get your ring with your D and C together, and this would have no centromere. So that would be an acentric. All right, so ring chromosomes can form acentric fragments, and then you have a karyotype of a female with a ring on an X chromosome would be a 46, so they still have 46 chromosomes. Sorry about that, quit that. And then you're a female, so you have the X, but one of your X chromosome forms a ring, so you put that R there to signify a ring, and then the other X has that ring structure. Now again, if the ring has a centromere, it can go through cell division, but it's often lost. So if centromere is present, it can go through cell division. If lost, then this will be a monosomy. And this can also be where you have mosaicism. Typically, your rings are going to form from chromosome 14 and 22. So high ring is associated with chromosomes 14 and 22. Now, ring chromosomes have been identified for every chromosome, but they're very rare and more common with respect to 14 and 22. Now we also have isochromosomes. Isochromosomes develop because there is incorrect division of your centromere. And so you can see that you have, in the case of a normal, you have your centromeres which are going to be replicated identically. And in the case of isochromosomes, the centromeres are off. And so most isochromosomes are going to be lethal. However, isochromosomes of X chromosomes are allowed, and they're going to have features similar to our Turner syndrome. So X chromosome is allowed. And your X chromosome will be XQ, and so the karyotype would be 46, X, and then you have an isochromosome, and it's on the Q arm of the X, and these are going to be similar to Turner. There's also been isochromosome on the chromosome 18q. And this is seen in Edwards syndrome. Okay, So now we're going to move into balanced rearrangements. And so again, these are where you have rearrangements, but you're not gaining or losing information. And a lot of these just mean that your genes are shuffling around, they're moving. So we have translocations. We have a reciprocal translocation and what's called a Robertsonian translocation. So in a reciprocal translocation, you're simply getting swapping of regions on different chromosomes. So here you're swapping this blue chromosome with this yellow chromosome, so you get partial information moved to each of the chromosomes. Robertsonian translocations look similar to a monosomy, except for you're not really losing too much information. It's typically with these acrocentric, so these with satellite chromosomes, and they fuse to form one chromosome. This gets trickier when the cells start to divide because now you only have one chromosome instead of the two. So with a reciprocal translocation, you're going to get a break in two different chromosomes, and the information gets mutually exchanged. So mutual exchange of material. And this is between two chromosomes. In this case, your carrier is usually going to be ineffective. You have in this picture here, you have a distal half of the short arm of C. So here's the distal half of the short arm of C, C6, chromosome 6, sorry, is going to move to the short arm of chromosome 3. So you have this guy that moves up here. And then the piece of chromosome 3 gets moved to chromosome 6. So this one goes here. So the translocation occurred, for example, we have a translocation that occurs at 3p13 and 6p14. Then the karyotype would be 46, XX, a T for translocation, and the translocation is between chromosomes 3 and 6, and the regions are P13 for 3 and P14 for 6. So this would be the karyotype of this individual. Now for a reciprocal translocation, the offspring can be normal. So our offspring can be normal, can carry the translocation, or can have duplications and deletions of material. So an offspring are going to receive the derivative of chromosome 3 and a normal 6, and therefore will be a partial trisomy of the distal portion of chromosome 6, for example. So if you have a offspring that has this derivative 3, so this goes into the offspring, and then you have, in addition to this, you have a normal 6. That means that this portion of the chromosome 3 is a duplication of this normal chromosome 6, and so this individual has a partial trisomy. Okay, so the next one is the Robertsonian translocation. So again, Robertsonian translocations are going to be where you have short arms, and these are of non-homologous chromosomes. So we have chromosome 13 and chromosome 14 shown here. They're different. And so the short arms are going to be lost and the long arms fused together. So we have short arms lost, and we have the long arms fuse. Okay, these are going to be limited to certain chromosomes, so only chromosomes 13, 14, 15, 21, and 22 can do this because these are acrocentric. Typically, you have no effect on the carrier. And that's because those short arms are very small and have no essential material. And your phenotype would then be unaffected. However, your karyotype would be changed. So your karyotype would be 45XX, for example. Because of the fusion of your 13 and 14, you're no longer 46. You now only have 45 chromosomes. So you have here another example of Robertsonian translocation. So this individual here would be a 45XY, if they're male. And then the translocation is you have a DER, so Robertsonian translocation 14, chromosome 14 and 21. And then the positions are Q10 on both. In this case, you're going to potentially have a cell that lacks one normal 14 and cells that lack one normal 21. And then the chromosome is, of course, the new chromosome is has fused the long arm of 14 and the long arm of 21. All right, so during meiosis, this translocated chromosome will pair with its homologs. So the 14 will pair with a normal chromosome 14, and the 21 will pair with a normal chromosome 21. If you in alternative segregation could produce offspring that are normal, or have a balanced translocation with a normal phenotype. If you're going to get an unbalanced segregation and your gametes are unbalanced, then this can lead to different outcomes. So you can have a trisomy 14. You can have a monosomy 14. Or a trisomy 21 or a monosomy 21. Okay, and so that's just shown through this diagram over here, where you have your normal, of course, and then you have your balanced translocation shown here, where you still have there's no loss of information here. This one you have a gain of information, so it's a trisomy, so you have 3 of the chromosome 21, so that would lead to Down syndrome. In this one you have a monosomy of chromosome 21, you have a trisomy of 14 or a monosomy of 14. So those are what can happen in your offspring. Now another chromosome change that can lead to a loss of information or a deletion or some sort of change of information is through an insertion. So This is showing an insertion with respect to chromosome 5 and chromosome 12, in which a portion of your chromosome 5 gets cut out and moved to chromosome 12. These insertions, of course, depend on where the cut are. Are you interrupting genes, etc.? But you're basically moving around the chromosomes. So for balance versus unbalanced, balanced insertion of one segment of one chromosome into a non-homologous other chromosome, and again, it depends on your coding versus non-coding and whether or not you are impacting genes. So for balance, you're going to have rearrangement of genetic material, no loss or gain, and this occurs, for example, when we have UV radiation. We get 2 breaks in our chromosomes, and then what happens is the fragment can turn 180 degrees and then religate itself. So that's what's shown here. It's hard to kind of see, but in this one we have A, B, C, D, E, and then we had some event, we'll say radiation, and so it broke the chromosomes, it broke them here, and so what happens afterwards is you had your fragment flip 180 degrees and then religate, so it religated opposite. So now you have E, D, and C. So basically it inverted itself, and so but it still is referred to as a balanced. Now inversions can cause 2 breaks on a chromosome, and then you can get fragments reinserted at the original site but inverted, just like I showed before. The thing to keep in mind here is nomenclature. So if it's going to include if it includes a centromere, so this is an inversion plus centromere, then this is called a paracentric inversion. Pericentric inversion. Inversion without a chromosome, or centromere, centromere is the para. So that's a paracentric inversion. So again, for here, we're including our centromeres, so you're getting some cuts and then some flipping. But the centromeres are included, so that is a pericentric. So the carriers typically have no phenotype. However, this can interfere with meiosis. And so these loops typically form during meiosis, and they can lead to duplications and deletions of regions of genes as well. Okay, so overall, we have chromosome abnormalities. So we've talked a little bit about the trisomies, the monosomies. It depends on what chromosomes are affected. There's only certain ones that are viable. You can have duplications, deletions. You can have changes in the actual content of the chromosomes that can be more deleterious than if you had a expansion or over expression of your genes from a gain of information, for example, so it's more tolerated. So with respect to morphologies and phenotypes, facial, cognitive impairments, growth delays are all common with these types of chromosome abnormalities.